Polymerase chain reaction or PCR, it is another important technique that is frequently used not only in molecular biology as well as in the field of genetic engineering or gene manipulation. So, what is its use? PCR can be used to amplify the target DNA. If we have very limited copies of our target DNA, then by repeated cycle with PCR, we can produce millions of copies of our gene of interest. What is the basic technique? So, here in a buffer, we can add our target DNA in the presence of primers and required nucleotides like adenine, guanine, cytosine, thymine. So, all the DNTPs are there. So, then the DNA is subjected to a denaturation step where the double stranded DNA it is converted into single stranded, and after denaturation then the hybridization step it is there where primers they will annealed at 5 or 3' ends, and after hybridization or annealing step then the temperature it is further lower, so that the next step, that is extension, it is there. So, by subjecting DNA to repeated cycles of denaturation, then hybridization and extension, we can produce millions of copies of target DNA within short period of time, like within 2 to 3 hours. So, it is having great application in the field of gene manipulation, where we can get number of copies of our target sequences. So, here this diagram showed theoretical PCR amplification of a target fragment. Suppose we have 1 or 2 fragments at the start, and from these fragments, after repeated cycle of denaturation, annealing and extension, after 30 cycles, that may take 2 to 3 hours, at the end we may have million of copies of the target DNA. Now, what are the applications of PCR? So, although we will discuss this topic, PCR amplification, as a separate chapter, but here you can note that it is very valuable technique to in prenatal diagnosis, where one can diagnose that either fetus having some sort of genetic disorder or not. So, we can take small tissue from the fetus and then we can check it by PCR. And one of the most important utilization nowadays, it is in the field of forensic science, where we can detect the history of some event, especially like in this case DNA profiling.